actually, also, Jung. I think number one, clearly the masking did not work. I think it's a masking thing, perhaps takes time to mature. And the same CD3 GFR bispecific also did not work, which was bought by Takeda. That was also a masking kind of a CD3 based T cell engager, and it has also, I understand, a lot of CRS. And but that molecule, this PSMA bispecific, does seem to have activity actually. That's interesting. I think for the solid tumors, the TCE is an interesting approach, and Amgen's DL3 CD3 bispecific, I think that's worthwhile to look at. Why the Amgen's CD3 DL3 worked well and did not have a severe CRS even at dose 100 milligram. Okay,
Yeah,
Amgen,
that's a big
why
question.
that right? Why that molecule is not masked, right? But it did
Right,
not have a
right.
lot of CRS either.
That's a
Yeah,
very good
I just
question.
want to emphasize, right, in pointing out J&J molecule and Amgen molecule, as well as the DLL CD3 molecule, these are not masked. You are right. And I think they utilize different antigens, and those antigens are more tumor specific, clearly. And I have done some background research as well. So you saw a recent Chinese product transaction, double epitope or paratope
Paratopic, yeah.
DLL3 CD3 engager acquired by AbbVie for $100 million upfront payment. So yeah, you need a good target that is clean, and you can go without masking.
Yeah, I think for Janice, there is one more thing which is most people believe is unique, which is the cleavage site also control the half-life. Supposedly the more you have very these are stabilized molecules have high half-life without cleavage, but in the tumor site cleavage activate, but the active molecule have much less hard half-life. That is something they believe generate much less side effect. I think from profile wise, cytokine profile wise, it does have a exciting data, but over time is still see some phase grade 3 toxicity, which might be possible, right? Different patient. So if they can control that, I think safety wise still I think it's interesting to me. But the question here is like whether this really drive efficacy in this specific disease and specific cancer cohort. We have to see more data. Then whether other design will be better. But DL3 is a little bit different story because very different tumor type and a very different targets. But I think someday we can summarize all this data and then give a complete summarize and discussion for what's a better T cell engager. All right, so over time, maybe like given time, maybe Yongjiang, you can start your second topic, which is very exciting to the field.
Yeah, it's actually related, but anyways, it's about multiple myeloma. It's quickly 1 minute that multiple myeloma, the treatment has evolved very, very significantly in the last 20 years or so. But from chemotherapy to this targeted therapy, including proteasome inhibitors and IMiDs, to now the antibodies, immunotherapy, and also CAR T. Next slide. So the treatment of multiple myeloma is really complicated, right? But if you look at it is really including the primary induction followed by maintenance. And when it's relapsed, you have a lot of relapse regimen, and finally it's salvaging therapy. So there are people who are eligible for transplantation, which about 35%. They would go through the stem cell transplant, and the other ones will go through chemotherapy and immunotherapy. So this is really, you can see, right? It's quite complicated with a lot of potential combinations from frontline to relapsed setting. So multiple myeloma is not a huge cancer, but it is incurable. However, the lifespan has prolonged so much, the patient would always need a new therapy. That's why this disease, although it is small, it has a very huge commercial potential. A lot of multi-billion-dollar drugs occur in this setting. Next slide. So this is immunotherapy for multiple myeloma, mainly including the CD38 antibodies, the CAR T, and the new antibodies such as the FR5H and also the GPRC5D kind of antibody target. And also here we're talking about a bispecific here. Next slide. So this is from J&J, and it is teclistamab, we call TEC. And this is a T-cell engager binding CD3 on one side and also BCMA on the other side. This is a mechanism. Next slide. So this molecule went through a phase 1/2 program, and it is very promising in the setting of relapsed refractory multiple myeloma. This was published in 2022, also in New England Journal of Medicine. You can see here the response rate on the left side, and also the DOR, the duration of response, the PFS, and all the other setting efficacy parameters are looking really promising for this bispecific molecule. It does have the CRS issue we mentioned before as a T-cell engager, but they did something called a step-up dosing, SUD, to manage this CRS, which is okay at some point. Next slide. So the trial design is shown here. It is a simple phase 3 study. Compare arm A, which is a TEC plus Dara, which is a CD38 antibody. And the comparator arm are the standard of care, which would be Dara subcutaneous plus the prednisone and also the dexamethasone pomalidomide, which is a proteasome inhibitor, plus the dexamethasone, or it's a DVD, which is Velcade, the proteasome inhibitor. So basically, it is the physician's choice on arm B, right? So it's either the DPD or DVD, and arm A, it is TEC plus Dara. Okay, both agents are from J&J. Next slide. So this is actually the dosing regimen, right? If you look at the bottom, the figure 2, it shows up here the narrow arrows are for TEC. So what it does is giving a day 2, giving a 0.03 milligram, a day 4, 0.6 milligram, and then on day 9, you give 1.5 milligram. So through this kind of a step-up dosing in the first week, it was able to reduce the incidence of CRS. And then you give a weekly regimen until cycle 2, right? So every week, then cycle 3 to 6, every 2 weeks, and cycle 7 and beyond, every 4 weeks. So by doing this kind of regimen, you're able to provide efficacy while reducing the incidence or frequency of CRS. Next slide. So these are results on the top left panel, which is small, but you can see the difference is huge. And the PFS curve on the top is the TEC plus Dara double regimen. The bottom is the standard of care DVP or DPD. And the PFS for the control arm is about 18 months. And for the treatment arm, it is not estimable because it did not reach the median yet. And the hazard ratio is very, very impressive at 0.17. So you're talking about an 83% reduction in the risk of disease progression or death. And this benefit is seen consistently across all subgroups. So they can, on the left side, the bottom left, the table shows you, or the forest plot shows you the benefit is very significant across all the subgroups. Okay, on the right table, it shows the overall response rate and also CR, a very good CR, and also very importantly, the MRD for this molecule, it was able to produce a higher overall response rate, higher CR, as well as MRD, the minimal residual disease, which is a measure of the 10⁻⁶, the positive myeloma cells, and that was a good indicator of long-term benefit. Next slide. So this is the overall survival. You can see here also, while it is immature, but the difference is quite small here as well. At 36 months, 3 years, the overall survival is at 83.3% for the Dara doublet, and it is 65% for the control arm, DPD or DVD. So this, again, you can see while it is still not mature, but the difference in the overall survival is quite clear. Next. So this is the safety profile of this molecule. You can see overall the adverse events include the hematological event, neutropenia, which was really similar between the two groups, maybe slightly higher for the doublet. In terms of the other like leukopenia, lymphopenia, they are also very similar between the two groups. So the safety-wise, there are no major issues. Although at the beginning, there are more patients who died on the Dara doublet because of infection, and the company implemented a very significant measure to provide the immunoglobulin to control the infection. So after that, there was a very, very similar death rate among the two groups. Next. So this is maybe a summary and a perspective on this program, which is, as I mentioned, this molecule really is the disease was pretty bad 25 years ago. Overall survival is only 2 to 3 years, but nowadays overall survival for multiple myeloma is about 10 to 15 years, so it's very, very long. Now the MajesTEC-3 trial, which is the one we just presented, it is the first phase 3 study in relapsed refractory multiple myeloma to demonstrate both PFS and OS superiority with the bispecific antibody, which is a T cell engager. Okay, and this is over and above the standard of care with antibody with IMiD or antibody and proteasome inhibitor triplets. What is really impressive was the magnitude of benefit, which is called a paradigm shifting and practicing changing. 83% 3-year PFS, which is more like a frontline therapy rather than a relapse refractory setting. So this trial really is going to push the community oncologist to adopt the T cell engager for multiple myeloma. And the KOL believed once the data become more mature and confirms the benefit, this doublet would become a new standard of care for early relapse. And this will also accelerate a broader move toward the introducing bispecifics earlier in the treatment course. So, and it also may open the doors to second line or even frontline combination exploring dual antibody immunotherapy. So I think this is a quite impressive results combining a T cell engager with the CD38 antibody, and which provides such a strong benefit, a 0.17 hazard ratio. So I think I'll stop here. This is just a quick summary of the results and the significance of this molecule.
So Yongjiang, this is really exciting. I have a question to you. So how this result and trial potentially affect the CAR T therapies? I have seen, like Legend Bio, the stock, right, has been affected. Like how do you think about the future of CAR T versus the bispecific given this trial?
Yes, that's a very good question. So the question is that the CAR T is using T cell to kill the myeloma cells, and this TCE is doing the same thing through this antibody. You can see here for patients who may not be able to get CAR T therapy, for example, a lot of patients actually got this. They may not be able to get reinfusion. This would be a very easy therapy because it's off the shelf, right? CAR T, you need a specific facility. It is much, much more expensive, perhaps $1 million really for CAR T therapy, which is reimbursed by Medicare. This is a much easier therapy. It's a sub-Q injection, so it can be used by the community oncologist, but CAR T cannot, right? You have to go to a specific facility to receive CAR T therapy. And eventually, I think this molecule perhaps would become, in my view, maybe used earlier than the CAR T therapy because of it's so simple to use compared to the CAR T.
Thank you. All right. So any other comments from the audience or from the panelists? There is one question say, why CD30 experienced patient only account for 5% of ITT?
Oh, when we do clinical studies, most of the time when you are using a regimen that contains a specific target, you would avoid the use of the same target in the frontline setting. But that is also a very good question because the CD38 has moved up to the frontline setting. The new standard of care for the frontline setting is the quadro therapy, which is the triple therapy plus the Dara or ISA, which are both CD38 specific antibodies. So in this particular study, this is certainly a good point about the patient who were on study may not have received the prior the Dara or other CD38 antibody.
So there's another question, like how does this compare to BCMA ADCs in second-line multiple
Yes,
myeloma?
from the data I've seen, I think it looks perhaps a bit better than the BCMA ADC. The BCMA ADC, as you have seen, it's pretty good actually, but it has a very severe ocular toxicity, right? Because almost all patients would have the eye problem, and some of them, many actually, have a very high grade 2 or 3 ocular toxicity, which is why the drug was pulled off the market for a while by the FDA. It just came back recently. It's got a reapproval after new studies have shown the benefit. So the ADC has a role as well, but I think this molecule, this doublet, does seem to have a better overall profile if you look at the efficacy and the safety.